Because fibrosis means the ECM becomes stiffer, and desmoplasia is just another word which also means fibrosis. And during fibrosis, the ECM becomes stiffer because there is more synthesis of collagen than higher than the degradation of collagen. So the synthesis of collagen 1 and 3 are increased, but collagen 4 is degraded. So fibrosis means the stiffening of ECM. So under normal conditions, there is a balance between degradation and synthesis, but during fibrosis, the fibroblast it decreases degradation and increases synthesis.
Hello,
That's
ma'am.
yes.
Yes, good evening. I think there is no one except me. I have joined recently because I've
Okay, okay,
come home from my university.
okay,
So,
sure, sure.
can you please from start from the first slide?
Okay, I will start with the first question.
Just a minute. Give me this.
You can type the answer in the chat box, then we can discuss it.
Okay.
Yeah, that is the correct answer. So, fibrosis means ECM stiffening. So, compared to a normal tissue, the fibrotic tissue will be stiffer. Even if you just touch it, you will be able to feel that it has become much more harder. So, the reason why it is becoming harder is that the fibroblasts, these are the cells which synthesize the extracellular matrix. And during fibrosis, they start to synthesize different types of collagen, and the rates of synthesis and degradation are also changed. So there is more synthesis compared to degradation. So that is why the extracellular matrix becomes more stiff. And desmoplasia is another word which also is the same meaning as fibrosis. And option three, it is not correct because it is given opposite. During fibrosis, collagen 1 and 3 are synthesized more, and collagen 4 is degraded. And option 4 is the correct answer. Fibrosis is associated with poor prognosis, meaning that if any cancer patient, so usually fibrosis is associated with cancer, the cancer tissue becomes more and more stiff as the cancer goes to a more and more advanced state. So the rate of recovery becomes lesser when there is fibrosis. So the fourth is the correct option. Let's go to the second question. So for this question, the answer is we'll go over all the options one by one. The first option is correct. Invadopodia are formed by cancer cells and they can degrade the ECM. So basically, the cancer cells they form these protrusions called invadopodia, and then they secrete matrix metalloproteases. So these are the proteins which can degrade the extracellular matrix, and it makes holes in the extracellular matrix, and the cancer cells can migrate through these holes, and this process leads to metastasis. That is, this is how cancer spreads from one organ to another organ. Option B, invadopodia and ECM degradation decrease with stiffness. Option 2 is not correct because many studies have shown that when the stiffness increases, the cancer cells are able to form more invadopodia and increase the ECM degradation, and there is more metastasis. So, option 2 is false. Option 3 is correct. Acini structures are formed in soft substrates. So these acini are basically hollow structures which are found in the breast. And the milk that is formed is stored in the inside of these hollow structures. And when scientists tried to grow these acini structures on soft and stiff, they were able to form acini only on the soft substrates. In the stiff substrates, there were cells inside also. The cavity was not hollow and the structure was also not spherical in shape. So option 3 is correct. It is formed only in soft substrates. It cannot be formed in stiff substrates. And option D, stiffness of healthy mammary epithelia is about 2000 Pascal. Option 4 is not correct because the stiffness of mammary epithelium is about 200 Pascal, not 2000 Pascal. So for this question, there are two correct answers, option 2 and option 4. So this is the study in which they have shown that in the softer substrates, we can see that like in about 170 Pascal, it has formed a nice spherical shaped ring, and in the middle there is a hollow hole. This is what is called the acini structure. But in the stiffer substrate, we don't see a hole. There are cells all around, and the shape is also not spherical. And this is another experiment in which we can see the invadopodia. So we can see where is the invadopodia with the actin mark. So these three dots. So they are the location where there is invadopodia. And in the same location, we can see these black dots in the Fitzy channel. So these dots are where the matrix protein has been degraded. So we can see that wherever there is invadopodia, the matrix protein has also been degraded. So from this, we can know that this invadopodia is like this kind of a protrusion structure which degrades the ECM, and the cell can then go through the gaps that are formed. So actually, the correct answer is option 2 and option 4, both of them. This is also correct. This one I'll just change the answer because both are correct. Or here also I can just change it. Let's go to the
Yes,
third
there
question.
could be printing mistake or the typing mistake, anything. No problem.
It's a typing mistake. From 2000, I have changed it to 200. Yeah, third one, the answer is correct. So when a tumor is growing, like many things will happen. First of all, we will get a lump-like structure. So that lump-like structure is because of an excessive growth of cells. And one more change is that the tissue will become very stiff compared to a normal tissue. And that is because the organization of the collagen will change and there will be more increased synthesis and deposition of collagen. and the fibers also, it will go from a more disordered structure to a more linear fibrous structure. And it is because of this collagen fiber and fibrosis that the cancer tissue becomes stiffer compared to a normal tissue. So the correct answer is option 2. Let's go to the fourth question. Yeah, fourth question also you are correct. So this Y27632, it is a drug and it acts on the ROROC pathway in the cells. And this ROROC pathway is what it phosphorylates the myosin and leads to myosin coming to its activated form. So when you treat the cells with this drug called Y27632, it will lead to the inhibition of ROROC activity and it will inhibit activated myosin. So it will not degrade the myosin, just that myosin will be there, but it will not get activated. So the cell will become very soft. Let's go to the fifth question. Yeah, so for fifth question, the answer is option one. When we get breast cancer, the tissue becomes stiffer because of fibrosis and increased collagen cross-linking. So it will lead to increased stiffness. So option two is correct. And option three is also not correct because the collagen cross-linking and stiffness, both of them will increase. And option four is also not correct because in the previous videos, we have seen that when the ECM stiffness is increasing, then the focal adhesions will also increase. So the correct answer to this is option A. When we get breast cancer, the collagen cross-linking will become more and the ECM stiffness will also become more. So because of the increased ECM stiffness, the focal adhesions will also become more. So the answer is option 1. Let's go to the sixth question.
Hello, ma'am. Can you hear me? Hello.
Hello, yeah.
Okay.
Yeah. I just came back.
Okay. The answer is three uniform. So
Yeah.
right.
Just give me a minute. Just 1 minute. Um, so this question is about how the blood flow inside arteries. So in a large arteries, if I'll show you the figure. Yeah, in the large arteries, um, near the wall of the artery, the fluid flow rate will be lesser because it is in contact with the walls of the artery. So there will be some kind of a friction effect. So the flow rate will be slower. But in the middle, it is not having any contact with any surface. So in the middle, the flow rate will be higher. So if you plot the speed, it will form a parabolic quadratic kind of an equation. And that is coming from the friction between the wall and the fluid, the blood flow. So the correct answer is option 2, quadratic. Is it clear?
Please explain it again.
Huh. So if this is the artery, this whole thing, this is the two walls of the artery. Then near the edges, so it is basically you can imagine a tube, and in the tube some fluid is flowing, and in the tube near the edge of the tube, so the fluid will be in contact with the wall of the tube. So because of that contact, there will be some friction between the blood and the artery wall. So because of the friction, the velocity will be lesser near the edge of the tube. But in the middle of the tube, it is not having any contact with any surface. So the velocity will be faster in the middle of the tube. So if you plot the velocity in the middle, it will be having the highest value. And both side, it will keep decreasing as we go from the middle of the tube to the edge of the tube. So if you draw it, it will give a parabolic shape. So that parabolic shape, it can be explained by a quadratic equation. So the answer is option 2, quadratic.
Okay. So parabolic shape. So that means y square equals to 4ax type, or y equals to ax plus bx square type, right?
Yeah, ax square plus bx plus c, that type.
No, not ax square. Y equals to ax plus bx square, or y square equals to 4ax, or x square equals to 4ay. These kind of, right?
Yeah. These kind of quadratic equations. Okay, let's go to the seventh question. The seventh question is, which mode of cell migration will cancer cell adopt in 3D collagen gel if MMP extracellular activity is inhibited? So cancer cell migration, it happens through two main modes. One is the mesenchymal migration and one is the amoeboidal migration. In mesenchymal migration, it is the normal migration where first there is a protrusion in the first, and then the protrusion is attached, and then there is an actomyosin force, this traction force because of the actomyosin contractility, and the back is detached. So in that way, the cell, it keeps moving forward. So for this kind of a migration, the cell needs space in front so that it can make this contact in the leading edge. The second kind of migration is the amoeboid mode, where in one part the cell will get contractile, and because of that contractile, the cell will be propelled forward. So in this amoeboid migration mode, the advantage is that the cell can squeeze in through the extracellular matrix. So depending on in which part this contraction is happening, the cell can be in different kind of shape. It does not have to maintain a front back shape like in the mesenchymal migration. Here it can keep any shape depending on the organization of the matrix. So wherever there is a gap, there it will make this bigger shape, and wherever there is no gap, it will contract, and this contraction itself will push the cell forward. So whenever there is no MMP activity, MMP is this protein which can degrade the ECM. So when there is no protein to degrade the ECM, it means that it has to find some gaps within the matrix to migrate. So only it can only do the amoeboidal migration, because in the mesenchymal migration, the cell cannot squeeze in. For example, if it is here and if there are any actin fibers in the front of the cell, then it cannot change its shape or anything. So the cell won't be able to migrate with the mesenchymal migration mode. So then it will switch to the amoeboidal migration mode. So the correct answer is option 3.
Okay, okay. It may be a little bit mistake.
Sorry?
Okay, actually, it should be amoeboidal migration because
Yes. Yeah.
as cell, a cancer cell adopt the 3D collagen gels. Right?
Yeah, it is in the 3D gel. Let's go to the 8th question. Yeah, this is a easy question only, and usually it is like very well established in the field that in the stiffer gels the cells can spread more compared to in softer cells they are more rounded up and the cell spreading is lesser. So the correct answer is option 2. More spreading on stiff gels and less spreading on soft gels. Let's go to the ninth question. So in this question, they are saying in which soft gel or stiff gel, in which of the gel the MMP secretion is high. MMP is again this protein which can degrade the extracellular matrix metalloprotease. And many studies have shown that in the stiffer substrate, the cancer cells are able to secrete more MMP. So the correct answer is option 3. Let's go to the last question for today. After this, we'll move on to the paper discussion from reading assignment. So in this question, there is a gradient gel, meaning that the stiffness is increasing from one end to the other end. So usually cells have this phenomenon called durotaxis. So durotaxis means the cells will go from a softer substrate to a stiffer substrate. So if there is a gradient, then the cells will, even if you have kept them, like initially seeded them on 100 Pascal, they will slowly migrate to the stiffer substrate and they will finally go to the 100 kilopascal end. So the correct answer is option 3. And this is happening because of durotaxis. Okay, let's go to the reading assignment. So this is the paper. We can, now the time is around 7:10.
Ma'am, please move back to the previous slide
This one? Okay, okay.
for 2 minutes.
Yeah, sure. I'll go to the next reading assignment.
Yes.
Okay. So this is the paper. I'll just put the DOI in the chat box. We can meet again by 7:35. Then we will have enough time to discuss it. You can read the paper from this link, not from the presentation. Because I just have the figures in the presentation. To read the paper, you can go to this link. I have typed it in the chat box.
Ma'am, you can discuss now.
Okay, we can start now. I'll just share the screen first. So this is the paper. Cell locomotion and focal adhesions are regulated by substrate flexibility. So this in this paper they have grown cells on polyacrylamide gels of different stiffness, soft and stiff gels. And it is a very important paper in mechanobiology because these days it is very like a very common technique to grow cells in different stiffness. And to do this, we use these polyacrylamide gels, and this is a very like one of the earliest papers in which they have used this technique. So when you make these polyacrylamide gels, how to control the stiffness of the substrate? So that depends on what is the percent of bisacrylamide. So basically, when you make these polyacrylamide gels, so you have to mix some things. The whatever you have to mix are, let me just type them. Yeah. So you have to mix acrylamide and bisacrylamide. So these are the main components in polyacrylamide gels. But if you just mix the two of them, the gel will not form. So it is basically a polymerization reaction. These acrylamide is the monomer, and this bisacrylamide is the cross-linking agent. And for this monomer, it has to go from the monomer to a dimer. So for that, we use something called APS and TEMED. So these two are, one is the free radical. So this, for this reaction to happen, there is a free radical formation. So whenever this free radical formation, it comes from the APS and TEMED. So whenever we add these two, there is this free radical formation, and because of the free radical formation, the acrylamide can go from its single monomer state to a polymer state. And this bisacrylamide is the cross-linking agent. Depending on this concentration, it will vary the stiffness of the gel. So if we add more bisacrylamide concentration, then the gel will become more and more stiff. So here we can see that when we increase the bisacrylamide, the Young's modulus or the stiffness of the gel is also increasing. And we are also able to see that the cell morphology is different in soft gels versus in stiff gels. So these are the main components that we add to make the gel. And depending on which stiffness gel it will change the mechanical properties of the cell. For example, the cell movement will be different. So here they have taken one point on the cell, like one focal adhesion, and they have tracked it every 10 minutes. And we see that in softer gels, it is a more like a more, there is a lot of variations in the movement. But in stiffer gels, the movement is very similar. Here there are many curves, but here the curve is more smooth and it is more single. And the speed is also higher. The cells are able to move easily in the softer substrate compared to the stiffer substrate. And then to then the authors were thinking that somehow the cells are able to sense whether they are on a soft substrate or whether they are on a stiff substrate. So to find out whether it is on soft or stiff, it has to use some proteins which are linked with integrin, because integrin is the cell substrate or the cell ECM protein. So this one important protein that is linked with the integrin is vinculin, and this vinculin is present in all the focal adhesions of the cells. So the authors, they have immunostained, they have used a technique called immunostaining, and they have done it in the softer substrate and then a stiffer substrate. So in the softer substrate, from the vinculin, basically we can see the focal adhesion, and we are able to see that the focal adhesions are like slightly bigger in size and they are more pointed in shape. Whereas in the softer substrate, the focal adhesion are like puncta, or they are more circular in shape and they are not that much organized. So the focal adhesions are bigger on the stiffer substrate compared to the softer substrates. And then phosphotyrosine. So tyrosine can exist in its phosphorylated form or in its unphosphorylated form, dephosphorylated form. And only in the when these proteins like vinculin are activated, then it goes from its dephosphorylated form to its phosphorylated form. So just to cross check whether these proteins are getting activated on the stiffer substrate, they also measured the levels of phosphotyrosine. And here we can see that in the stiffer gels, the levels of this phosphotyrosine is much more higher. So from this, we can say that the in stiffer substrate, there is more phosphorylation and there is more activation of these proteins in the focal adhesion like vinculin and paxillin. So this was immunostaining, meaning that the cells are fixed. So the next experiment they did it for live cells. They tracked it every 10 minutes, and then they also used certain drugs. So the first drug they used was PAO. So basically, it will not allow the tyrosine to be phosphorylated. So when the tyrosine is not phosphorylated, the cells are behaving the same in soft and stiff substrate both. Because then, so for only if the tyrosine phosphorylation is happening, then it can go differentiate between the stiff and soft substrate. But now, because this phosphorylation is not happening, we see that the vinculin distribution is similar in stiff substrate as well as in soft substrate. And then they also used an inhibitor of myosin motor. So we in many studies it has been shown that the cells have to apply a force to sense whether it is on a soft substrate or stiff substrate. But when we inhibit the myosin motor, the cell is not able to apply any force. So again, it is not able to differentiate between soft and stiff. So the basically the force applied by the cell will become very less when we use this drug. And because the cell is applying less force, it will also feel a lesser force from the substrate. So the cell will think that it is in a soft substrate. Even though it is in a stiff substrate, we can see that the vinculin and the phosphotyrosine distribution, it is similar to a soft substrate because the cell is not able to apply more force to the substrate. So that is what is the main concept in this paper. They have grown cells on different substrate and they have seen what is happening to the different proteins in the cell. And it is very important because it is one of the first papers in which they have used these polyacrylamide gels to grow cells. So, and one more thing that we do in the lab is once this gel has formed, the cells cannot bind directly because the cells will need a extracellular matrix to bind. So first we will make the gels, then we will do an ECM coating like fibronectin or collagen. And after doing this only we will seed the cells. Otherwise, if you seed the cells without the fibronectin coating step or the collagen coating step, the cells will not stick to the gel. They will just float and they will eventually die. And this paper is also important because one is we can control the stiffness, and PAA gels are very good because it is transparent. So we can use in microscopy. And one more important thing is that this is the gel that we use for traction force microscopy. So in traction force microscopy, when we make the gels, we do the same things, but we add one extra component, fluorescent beads. So this we add only if we want to do TFM or traction force microscopy. So that is why it is a very, very important paper in this field. So this gel, it is the same gel that we use in SDS page, just that the stiffness and the thickness are different. This PAA gel, it is what is used in SDS page as well. Any doubts or any questions?
So the PA gels increase the stiffness. Right.
Can you repeat? I didn't hear you properly.
So the PA gels increase the stiffness of the
Yeah, we
cell.
can make, yeah, depending on our experiment, we can do different stiffness gel. Like in these experiments, in these questions, we were discussing, right? When we do 100 Pascal to 100 kilopascal. So these kind of experiments to do, we have to use the PA polyacrylamide gels. And in the stem cell also, we were saying that they have done experiments where in the soft gels, the cells can become more neuronal fate, but in the stiffer gels, they become bone cell fate. So those experiments also they have done using these polyacrylamide gels. Because you just have to change the amount of bisacrylamide and you can change the stiffness of the gel.
We have to change the amount of bisacrylamide and then change the amount of polyacrylamide gels, right?
Yeah, if you change the bisacrylamide, then if you make it more, then the gel will become more stiff.
Okay. Thank you.
Yeah. Any other questions? Seventh week lectures you have started watching?
Not still. No.
Okay, okay. I think this will come in more detail in the next week. This traction force microscopy and other experimental techniques.
The official admit card of the examination is not released still yet, right?
Yeah, it is not yet released. I think they will give it last time I remember, I think 1 or 2 weeks back it was released. I think so. And then they will ask you for exam center also. That I think that also they have not yet asked, right?
Hmm. Okay.
Yes.
And
First you have
exam
to
will be on
prepare.
the 24th September, Sunday.
Okay. Okay. Oh, okay. You have done any other NPTEL course before this?
Yes, previously I had done one NPTEL course regarding structural biology, instructed by
Hmm.
Professor Shougoto Hajra, IIT Roorkee
Oh,
professor.
okay. Okay. So mostly the exam pattern and everything will be similar only. Like for all the NPTEL courses, the assignments will be mostly MCQ type and the final exam also. So it will be like the same pattern only. What is your background, your biology background?
I mean, hello, ma'am. Are you there?
Yes, yes, yes.
Okay. Actually, now I am a UG student. I am pursuing BSc Biotechnology degree in Maulana Abul Kalam Azad University of Technology, Horinghata, Kollani. Now I am in the final year.
Okay, okay, I see. And you are planning to do research after this? Like go into research or what is?
It's yeah, it will up to the situation of the job openings and other things that which I can prefer, that academics or industry or any other thing. I have to boost up my CV for my upcoming career.
Yes, yes, yes.
This is very
That's what
vital.
I was asking because you are doing this course, right? This is more of a research oriented course. Like not many industries are there in this field. So that's why I was thinking that maybe you're more interested into research.
Actually, I'm doing these kind of courses like which are not in our college syllabus
Yes, yes.
for extra knowledge development and
Okay.
other things.
Yeah, yeah, that is good. That will, if you put these things in your CV, it will be very helpful.
Hmm.
You have
And
any? Yeah,
and
yeah.
in our last semester, we have to go for the dissertation the outside. So CV is very vital to show
Yes.
in the to get good opportunity for dissertation in good institution laboratory.
Hmm.
You know well better than me.
So you have started applying for thesis?
I'm still not now, but I have to mail severe faculty members of different institutes.
Yes, yes. So if you, because you have done this course, you can try to email professors who are working in this course, and you can mention that you have done this course on NPTEL if you are finding it interesting.
Okay, ma'am. So thank you for today.
Yeah, yeah. If you don't have any other questions, we can end the session today.
Yes.
Okay. See you next week.